Hello and welcome to some biology class. Friends, today we shall discuss about zinc finger nucleases. Zinc finger nucleases are artificial restriction enzymes. They are the engineered proteins generated by fusing a zinc finger domain to the cleavage domain of FOC1 restriction endonuclease. These were the first targeted nucleases to achieve widespread use. Zinc finger nucleases function as dimers. Each monomer recognizes a specific half-side sequence, typically 9 to 18 base pair of DNA via the zinc finger DNA binding domain. Dimerization of zinc finger nuclease proteins is mediated by the FOC1 cleavage domain, which cuts the DNA within a 5 to 7 base pair spacer sequence that separates the two flanking zinc finger DNA binding sites. So each monomer of zinc finger nuclease consists of two domains. One is a zinc finger DNA binding domain and the other is a cleavage domain of FOC1 restriction endonuclease. These two domains are fused with the help of a linker peptide to form a monomer. In this figure, this is a double-stranded DNA molecule. On that side, this is a monomer of zinc finger nuclease. And on this side, this is another monomer. Both of them constitute a dimer, and each monomer consists of a zinc finger DNA binding domain and the cleavage domain of FOC1 restriction endonuclease. The function of zinc finger domain is to bind with specific sequence of DNA, and the function of FOC1 cleavage domain is to cut the DNA at a specific sequence. Zinc finger DNA binding domains are derived from zinc finger proteins. Zinc finger proteins are one of the most abundant group of proteins with a wide range of molecular functions. The DNA binding domain of individual zinc finger nuclease typically contains between 3 and 6 zinc fingers. In this figure, there are 5 zinc fingers. Each zinc finger consists of approximately 30 amino acid residues that are organized in a beta-beta-alpha motif that includes 2 cysteine and 2 histidine residues that remain associated with the zinc ion. The amino acid residues that facilitate the recognition of DNA are located within the alpha helical domain that typically interact with a 3 base pair of DNA with an occasional overlap from an adjacent domain. The zinc finger domain can be engineered in order to target specific base sequence within a complex genome. Using methods such as phase display, a large number of zinc finger domains recognizing distinct DNA triplets can be identified. All these domains can be fused together to generate polydactyl zinc finger proteins which can target a wide range of possible DNA sequences. A major concern associated with the use of zinc finger nuclease in genome editing is off-target mutations. Unlike CRISPR, Cas9 and TALENs, difficulties associated with the construction of zinc finger arrays have hindered their widespread adoption in unspecialized laboratories. In particular, it remains challenging to create zinc finger domains that can effectively recognize all DNA triplets. Nevertheless, the potential for zinc finger nucleases to mediate specific and efficient genome editing is evidenced by the ongoing clinical trials based on zinc finger nuclease mediated knockout of HIV-1 co-receptor CCR5 to treat HIV and AIDS, and another ongoing trial which is based on site-specific integration of factor 9 gene into the albumin locus in order to treat hemophilia B. So friends, this is all about zinc finger nuclease. For more updates, please subscribe my YouTube channel, Sam Biology Class.